Hello friends, welcome back to my YouTube channel Nasrin Badasale. In this video, I'm going to tell you top 50 important topics for your GATE 2024 biotechnology paper. So first thing is regarding to enzymes that are used in rDNA technology with special focus for restriction and modification enzyme and end modification enzyme. So you can refer analysis of genes and genomes by Rees book. Second is vector. Again, you can refer the same book. So vector is very important. Generally, they will be asking you the size, insert size. Okay, and also with regarding to uses, utility of those vector. Next is PCR. So you need to know the general principle of PCR, followed by that numerical sums with regarding to PCR. Okay, so if you go to YouTube, you can find many YouTube videos starting from Somus Biology to Unacademy. Many YouTube channels are providing numericals with regarding to PCR. And next thing is regarding to types of PCR, special focus to reverse transcription PCR and real-time quantitative PCR analysis. Okay, so whenever you are searching about any topic, type the topic in YouTube, and additionally you need to add Unacademy. So in Unacademy, they are providing some more good content with regarding to various techniques and also for some other topics also. Okay. Next is molecular marker. So you need to know about list of dominant marker and codominant marker, and also about the DNA fingerprinting. Okay, you need to know about RFLP, AFLP, RAPD, like that. Okay, molecular marker also YouTube videos is available in not in my channel. So some other channel they are placing some good content with regarding to molecular markers. So my channel basically concentrated on providing revision series. So I'll be starting those revision series 2 months before examination. So if you are focused on those revision series, or if you want to cover up the GATE biotech syllabus for last minute, you can follow my channel. Next is labeling technique for RNA and DNA. So this include radioactive labeling and non-radioactive labeling. Next is DNA sequencing. Starting from Maxim Gilbert till next generation sequencing, you need to study about the DNA sequencing. So for the technique, you can refer another YouTube channel that's called as BAAYO, given by R.T. Radimam. Okay, next blotting, northern blotting, southern blotting, western blotting. Next is mutagenesis, site-directed mutagenesis, followed by that CRISPR-Cas system. So next thing is bioinformatics. So bioinformatics is very important, and those questions are easy for us to solve. So first go through the previous year question as asked in bioinformatics, or else if you simply type bioinformatics for GATE, BT or GATE Excel, again YouTube channels are providing those content. In my channel, I had placed a separate video for bioinformatics for ICMR examination and GATE examination. Okay, so you need to know about different database, sequence database, name alone you need to know. Okay, structure database, BLAST, FASTA, sequence file format, and scoring matrices, and pairwise alignment, multiple sequence alignment, like that you need to know about all those things. Next is microscopy, all type of microscopy. Starting from light microscopy till 10 you need to know, and again numericals for microscopy you need to study about the numerics or problems from microscopy. Next is spectroscopy. This include UV visible, CD, IR, fluorescent. Generally they will be asking match the following type of questions only. So you need to know about the principle and the function application. Okay, next is electrophoretic technique. This includes STS page and agarose gel electrophoresis, followed by the gene expression techniques. This include microarray. Next is flow cytometry is very important, and fluorescent activity cell sorting is also very important. Next is ELISA technique. These are immunological technique. So you can able to find a separate YouTube videos regarding to immunotechniques. Okay, those YouTube videos will be placed for CSAR unit number 30. So you can follow those videos for your GATE examination also. Followed by that chromatin immunoprecipitation sequencing. Next is regarding to bioprocess. If you are a MSc biotechnology, if you can't able to cover up the entire section 4 and section 5, you can concentrate on the important topic that I am going to tell you. So you need to study about the production of biomass, production of primary metabolite, secondary metabolite, production of biofuels, bioplastic, industrial enzyme, and how antibiotics are produced in industrial scale. Next, large scale production and purification of recombinant protein and recombinant metabolites. These are important. Next is hybridoma technology. Next is animal cloning, transgenic animal, knockout animal, knock-in animal. So 4 techniques are highly important. Knockout is very important. So you need to study about the selection of animal, like selection of recombinant. Okay. So you need to study about neomycin resistant cassette and thymidine kinase sensitivity. Okay. So yeah, transgenic animal, you need to know about the methods, and also if there is any new transgenic animal, or you need to study about the list of transgenic animal and about the application. Next is culture media composition and growth condition. This is with regarding to animal cell culture. So while studying transgenic animal, you also need to know methods of gene transfer in animal. Next is plant growth regulator and production of secondary metabolite, and those application like list of secondary metabolite and their medicinal properties. And also you need to know what plant product of industrial important and artificial seeds, protoplast, isolation of protoplast and protoplast fusion. Next, same transgenic plant and gene transfer in plants. For this transgenic technology and all, you can refer the analysis of genes and genomes, a book that's well sufficient. Next is selection marker and reporter gene. Next is chloroplast, that is plastic transformation technique. Next is this upstream and downstream processing is very important. So you can refer Biotechnology by Satyanarayana book. Okay, so you need to study all like filtration techniques, centrifugation techniques is very important, and chromatography is highly important. Okay, next is DNA damage and repair technique, DNA repair diseases, RNAi technique, and also you need to study about the replication, transcription, translation. So for which you can refer Living Genes book. Okay, additionally you also need to know about structure of DNA, RNA, and also about general concepts of nucleic acid. You need to study about Chargaff rule and how you will be analyzing the melting temperature of DNA, DNA denaturation, renaturation kinetics, stability of DNA, all those things coming under the mol bio. Next, about cell structure of prokaryote and eukaryote. So there will be a tabulated form will be available. Prokaryotes, eukaryotes, and archaea. So don't forget to study those things. Next is cell cycle and also control of cell cycle. Additionally, you need to know about cell-to-cell communication, cell signaling, post-translational modification is highly important, friends. Next is about the protein transport across the various cellular organelles. Next is apoptosis, cancer, and autophagy. Next is ECM. For which, for cell biology, you can refer Albert book alone. No need to go for referring Karp or Cooper. Just refer Albert book. Followed by that, in evolution, you need to concentrate about speciation, adaptive evolution, neutral evolution, and genetic drift. And also genetics, you need to study about epistasis, extra chromosomal inheritance, and microbial genetics, which include conjugation, transduction, transformation. Okay, and you need to also know about horizontal gene transfer and gene mapping, and also you need to know about transposable element. Okay, next you need to study about chromosomal variation. So for genetics and all, you can refer Benjamin book, that's well good, and also most commonly questions are asking from that book only. Next is about genetic disorder, both the single gene disorder and chromosomal disorder. Next in immunology, you need to concentrate on antibody structure, types, and their function. Next is antigen antibody reactions like precipitation, agglutination reaction, complement system, and also primary lymphoid organelle, secondary lymphoid organelle, major histocompatibility complex, antigen processing and presentation is very important, and about difference between polyclonal and monoclonal antibody. Next is hypersensitivity, four type of hypersensitivity reaction. Okay, next is regarding to autoimmunity. So for immunology, if you can't able to refer Cube book, you can refer Richard Kaiko Essential of Immunology book. So don't worry, I'll be attaching a link of those book below. So okay, next is graft versus host reaction. So transplantation is very important. Okay, so about immunization, like active immunization, passive immunization, natural immunization, and artificial immunization, about vaccine and their type. Okay, next is microbial interactions, and with regarding to microbiology, you need to concentrate on microbial interaction, virus structure and classification of virus, various methods in microbiology, coach postulate, for which you can refer Prescott alone. Next is microbial growth kinetics and microbial nutrition. You need to study about all those things. Next, you need to cover up microbial media like that, and also in microbiology also you need to cover up microbiology problems. Okay, there will be various problems problems coming from microbial growth and nutrition. So kindly do study all those things. Next is nitrogen fixation. Next, you need to cover up microbial diseases. Not only disease caused by bacteria, you need to concentrate on bacterial disease, fungal disease, protozoal disease, all those things. And also you need to study about host and pathogen interaction. Next is about antibiotics. So antibiotics is highly important and antimicrobial resistant. Next is biological membranes is very important. So in microbiology, you also need to concentrate on sterilization techniques also. So in cell biology, you need to study about biological membrane transport across biological membrane. Okay, next is electron transport chain, an inhibitor of electron transport chain. Of course, in biochemistry, metabolism is very, very important. Okay, next you need to study about enzymes, which include classification of enzyme, enzyme inhibition, enzyme kinetic, and various problems. Next is biosynthesis of various, like you need to study about different biological pathway happening, their location, and what about the net yield, how many ATPs produced in glycolysis, and also all those things. So you need to study about the location of biochemical pathway in plant prokaryotes and eukaryotes. Next is peptide digestion or peptide sequencing technique. Next is antibiotic, as I mentioned you earlier. Next is Chagaf rule. Next about the DNA, RNA, and different forms of DNA like aDNA, bDNA, zDNA. Next is about Ames test and operon concept, lab, trip, and ARC. These are very important. And about pedigree analysis. Okay, so both theory and problem of pedigree analysis you need to cover. So these are the important topics. So first concentrate on these important topic and then go. Okay, so thank you friends. Thanks for watching this video.